Okay, let's get started. Welcome, my friends, to the inaugural College of Environment Postdoc Pecha Kucha. Thank you so much for coming. I can see already a big number of us here, I think over 100 right now. Even when it's sunny outside, as you can see in my window, pretty awesome outside and unusual. So my name is José Guzmán, and I'm assistant teaching professor in the School of Aquatic and Fisheries Sciences and in the Marine Biology program. And I'm co-hosting this event with my esteemed colleagues, Bruce, Nelson, and Julia Parrish. Bruce, Julia, you want to introduce yourself?
Sure, I'll introduce myself. I'm Bruce Nelson. I am the Associate Dean for Research for the College of the Environment, but on my normal days, I'm a professor in the Department of Earth and Space Sciences, and I focus on geochemistry, volcanology, and transport of toxic metals through our environment. This is my second tour of duty as Associate Dean, and it's a real privilege to have the college-wide perspective of what goes on. It's an amazing place. So I hope I get a chance to talk with all of you in more detail after this.
Excellent. And hi, everybody. I'm Julia Parrish. I am the Associate Dean for Academic Affairs in the College of the Environment, also a professor in the School of Aquatic and Fisheries Sciences and in the biology department over in Arts and Sciences. It's also my privilege to co-host with Jose and Bruce. So just a few housekeeping details before we get going. Attendees, thank you for joining us today. If there are logistical or technical issues, can you please put them in the chat? And please reserve the Q&A, which is that little icon all the way over to the right at the bottom of your screen for questions for our postdocs. They will be giving 5-minute pre-timed talks, so fast and snappy. I would love for you to cheer them on in the chat and in the Q&A. And once they finish, they are waiting for your questions in the Q&A. So please type them in fast and furious, and you can engage in a conversation with the postdocs. We'll be wrapping up by having some little bit of Q&A at the end, but please do not hesitate to send your questions to the postdocs as soon as they have finished. Okay. I think we're ready to go. I'm going to turn my video off and let Jose do the introduction.
Okay, so the first one, we have Stacy Amergy from the School of Aquatic and Fisheries Sciences. UDA postdoc for a year now, and her advisor is Sarah Converse. Stacy uses quantitative modeling methods to understand the ecology of natural systems and to provide information to management partners for the conservation of wildlife species. The presentation today is Silent Islands, the quest to manage brown tree snakes. Stacy, you ready?
I am ready.
Go for it.
Awesome. Imagine yourself in this landscape looking out at the tropical forest. Think of the sights and the sounds. Now imagine that this landscape is utterly quiet except for the sounds of the waves crashing on the beach below. This tropical yet silent island is on an island way out in the middle of the Pacific Ocean, about 6,000 miles or 9,000 kilometers away from where I and many of you are currently sitting in Seattle. The silent island we're talking about today is in a chain of islands called the Marianas, with everything above that red line that you see being part of the Commonwealth of the Marianas, and everything below that line being a US territory. And specifically, the island we're discussing is Guam. Guam is the ancestral and current home of the Chamorro people, but there are also several large US military bases on the island and a population of nearly 170,000 people. And like you imagined at the beginning, this tropical island used to be teeming with birdsong and life. These are just specifically the forest birds of Guam, but there used to be a variety of coastal, grassland, and marsh bird species on the island as well. But that is no longer the case. The birds highlighted in the red box are extirpated on Guam, meaning they no longer occur on the island, and in some cases, they no longer occur anywhere in the wild or in captivity. And that disappearance can largely be attributed to this guy. This is the brown tree snake, which is an invasive species on Guam. Brought to Guam by accident in the 1940s, the number of snakes absolutely exploded in size after their introduction. The brown tree snake is largely arboreal and nocturnal, and that means that it is really difficult for people to find in a tropical jungle. By the time that people realized what was happening with the brown tree snake, the damage was done and the solutions for removing it were no longer simple. Normally, the brown tree snake is found in Australia and Papua New Guinea, but the snake was now on an island with none of its usual predators and basically an open buffet of animals that had no idea what a snake was. There are no native snakes on Guam. And this worked in the brown tree snake's advantage. Additionally, the brown tree snake is venomous, only mildly to humans, but the venom is particularly effective on birds and lizards. When the birds roosted at night, the snake could come in and eat or just simply bite and kill individuals. And this has had a lasting effect on the ecosystem of Guam. Not only have the snakes directly killed the birds, lizards, and finihi, which are the Mariana fruit bats, but these species are part of a much larger network of other species. A lack of pollinators and seed dispersers on the island means that the forests no longer regenerate the way they should, and with no birds to eat them, the insect and spider populations are many times that found on other islands. So, how do we fight back? Well, there are really dedicated folks using multiple tools to catch and study this snake, as you can see here, which increases our understanding of potential techniques for the monitoring, removal, and management of this species across its life cycle. And we want to move forward, and we want to move forward in a way that will allow us to hopefully bring native species back to the island of Guam. And with all this knowledge, we now have so many pieces that are key to this management puzzle, but we need to figure out how the pieces actually fit together. And so that's where I come into this scenario. I'm taking all the different pieces of data from all these different monitoring methods and figuring out how to piece them together into a monitoring plan that will allow us to work to get what we need with the money that we have to actually do it. And so, given that I know so much about how the snakes move and at what rate we catch them by hand or in traps or at cameras, I can simulate snakes, seen here as the little brown dots, moving around a landscape with different monitoring scenarios, like these red traps that rotate around on different trapping nights. And by trying out these different scenarios through simulations, I can see what combination of tools and for how long we need to place them on the landscape to get an idea of how many snakes there are in an area where we're interested in reintroducing native species. And I want to note that conservation does not occur in a vacuum, and to get a task done, it requires money. But by explicitly testing these different combinations and trying them on for size, so to speak, we can be efficient and effective in the kinds of options we recommend to managers. With the ultimate goal being to reintroduce these species that still exist. These birds are not just drawings, but are waiting in captive breeding facilities or on other islands for their potential reintroduction. Creating an optimal plan forward will allow us to ultimately restore ecosystem function to Guam. So as I said earlier, conservation does not happen in a vacuum, and I want to acknowledge all the wonderful people who contribute to this project day to day on working to make Guam a conserved, restored, and revitalized landscape. And thank you so much for your time today.
All right, beautiful. Thank you for that. I love that. Conservation does not occur in a vacuum. I can only imagine the lessons that islands all across the Pacific are going to draw from your work. Thank you very much for that, Stacy. That was excellent. I would like to turn next to Ellie Bors. She is a postdoc working in the Cooperative Institute for Climate Ocean Ecosystem Studies. Ellie is a molecular biologist, and she studies the population genetics of climate-driven distributional shifts. She's working with Lawrence Hauser and SAFS and Ingrid Spice, who's in the NOAA Alaska Marine Science Center. And Ellie's presentation today is entitled Managing Fish on the Move in Norway's Arctic. And Ellie, are you ready?
I am ready.
Okay, let's go.
Think about any species on the planet, and then think about where you could find that species, its range. Within its range, how is it distributed? You'd be wrong to think that that distribution is static. No matter what kind of species you are thinking of, its distribution is not a constant. Some species migrate and others may experience changes in distribution for a variety of reasons. One major reason is climate change and the associated changes in environment. What happens when the change in climate also changes the distribution of our food? There are many consequences and factors to think about related to distributional shifts, and at the UW, I'm focused on genetic questions. But today I'm going to talk to you about an opportunity I had as a Fulbright Arctic Initiative scholar to study the policy and management context of distributional shifts. The Fulbright Arctic Initiative brings together researchers from all 8 Arctic Council nations and enables scholars to do collaborative research and go on research exchanges. I did my 10-week research exchange in Tromsø, Norway. It's a beautiful small city with shockingly good infrastructure and excellent views. This was the view from my bus stop. I was hosted by researchers at the Arctic University of Norway. Tromsø is a city on an island. Like most of the coast of Norway, it is defined by inlets, fjords, mountains, miles of coastline, and fishing. The stunning Norwegian coastline folds alongside the Norwegian Exclusive Economic Zone, which spans from North Atlantic waters to Arctic waters and includes many shared boundaries with other countries and with international waters. Depending on who you ask, Norwegian waters also include many areas of contested sovereignty, but I won't dwell on that too much today. So back to fishing. The coast is dotted with small fishing villages like this one in the Lofoten Islands, where fleets of boats fish in coastal waters. Farther north, larger ships fish in the open ocean of the Barents Sea. The Lofoten Islands are historic Viking land, and many have fished cod here for over 900 years, drying them much like you see today in large racks along the coastline in this picture. Fishing is integral to Norwegian culture, which is illustrated most clearly through drawings I saw by middle school students hanging in a classroom at the Fisheries College. The detail of the artwork in the upper left shows a whole supply chain, from a larger commercial vessel to a fish market and cafe, and other students chose to depict an ecosystem approach to fisheries. Now I will focus on what's going on in the Barents Sea to the north of Norway, which is home to several iconic and lucrative fisheries. The Barents Sea is an ecological transition zone with Atlantic or boreal fish communities in the southwest and Arctic species in the northeast. Data document a spatial shift in fish communities to the east and the north. These shifts may not look dramatic, but they indicate a potentially serious movement of commercial fish species away from ports and across maritime boundaries. During my research exchange, I asked some big questions. How might distributional shifts affect Norwegian fishing? What is the management context? And where does the science fit in? In order to learn about these questions, I convened a workshop of 8 experts. My hosts, Raul and Michaela, participated, along with other ecologists, economists, and international policy experts. We ate some waffles, and they were, of course, delicious. Our conversation focused on the annual regulatory cycle, the process by which fisheries are managed in Norway. In this image, starting at the top, each June, Norway receives catch recommendations from an international body. In the fall, it negotiates over 20 international agreements. The government holds a stakeholder meeting in the late fall, which is followed by proposed regulations that are approved and implemented in January. Fishing and science contribute data to the process. Science about distributional shifts could be incorporated at many points of this cycle, and today I will highlight 2, catch recommendations and international negotiations. Catch recommendations, as well as large ecosystem reports, are provided by the International Council for the Exploration of the Sea, or ICES. Strategic approaches that link single stock advice to long-term ecosystem trends may improve adaptation efforts in the face of distributional shifts. Furthermore, Norway has a strong track record of cooperation with Russia in the Barents Sea. Annual fisheries surveys, data sharing, and negotiations are facilitated by the Joint Russian-Norwegian Fishing Commission. Established institutions for international cooperation like this one will enable responses to distributional shifts across boundaries. International institutions will be important as nations learn to manage living resources that are on the move. By using Norway as a case study, we can find hints about how fisheries can adapt worldwide and how science can play a part. So I will close with another piece of art from one of Tromsø's middle school artists and fish enthusiasts. Thank you very much for your time.
Come on. Wonderful. Oh, Ellie, that was great. I mean, it's really interesting to think about how Norway can be a training ground for how we're thinking about managing fisheries and marine resources more broadly as they literally move through static political boundaries. Super interesting concept. Thank you so much for that. So now we're going to turn to our third postdoc, Simone Desroches. Simone, hi. Simone joins us from the School of Aquatic and Fisheries Sciences, where she's been a postdoc for about 1.5 years. She studies how diversity within species is shaped by anthropogenic and natural forces, and how this affects ecosystems and people, working with Jason Toft of the Wetland Ecosystem Team. And today, her presentation is Social Variation Within Humans Shapes Our Interactions with Other Species. Simone, are you ready?
Sure am.
Great.
Thanks, Julia. Okay, often we think of biodiversity as the number of different species. If you consider that each one of these species has just one uniform function, then an ecosystem with more species will have more diverse functions. But there's also a lot of diversity within species, which we call intraspecific variation. And this can be variation in morphology, what individuals look like, their behavior, and multiple other traits. This means that one species does not always have just one uniform ecological function. So that in fact, variation within one species means they can have diverse effects in an ecosystem. For example, experiments show that variation in mouse size within one species of fish affects, of course, the size of food that they eat. So that fish with smaller mouths eat smaller invertebrates, and of course, fish with larger mouths eat larger invertebrates. This thus affects the different ecological responses, like the composition of large and small-sized invertebrates in the community. This ultimately even affects different food webs. So we've looked at a bunch of studies on different species. So in this figure here, each point is the average ecological response in one study, with larger intraspecific effects on the left and larger species effects on the right. And so you can see by all the points in the middle, the effect of substituting different forms of the same species was often similar to the effect of substituting entirely different species altogether. Now, some species have especially large effects and can even shape entire ecosystems. We call these keystone species or dominant species or even ecosystem engineers. And we'd expect that variation within these species to be really important for ecosystems. So humans are often considered the most important and dominant ecosystem engineers. Our own activities, our behaviors, our needs and wants can drive the ecology and evolution of many species. Cities are a great example of how we create and modify habitats, so they're drastically different from the way they were before. We do this in a way that generally benefits us, but these new habitats obviously cause extreme ecosystem level changes, yay, that indirectly alters the ecology and evolution of the species that live within them. We can also directly co-evolve and modify other species that live close to us, like pets, farm animals, cultivars, and pests, through processes like artificial selection and even genetic modification, or more simply, domestication, selective harvest, and pesticide use. So, for example, we have indirectly driven evolution in stickleback fish in California through climate change, and we've directly caused evolution of smaller salmon through selective harvest in Alaska. But just as with other species, humans do not have just one uniform ecological function, and we show substantial variation in our species. And of course, some of this diversity is genetic, but it's often non-genetic as well. And it manifests in the variation in how we behave and interact with our surroundings. More so than perhaps any other species, our variation is strongly influenced by different cultures, traditions, trends, values, and even systems of government. In fact, we even regulate the activity of other humans. Sometimes this is negatively imposing through racist policies like redlining in our cities. And these actions cause heterogeneity or a variation in our landscape, ultimately influencing the ecology and evolution among cities. For example, access to resources for waste disposal may be more limited in lower income neighborhoods, leading to better mosquito habitat and more mosquitoes in these neighborhoods. This disproportionately burdens people in those lower income neighborhoods, driving localized increased pesticide use and ultimately evolved pesticide resistance in specific locations. This is an example of socio-eco-evo dynamics, which I'd argue are crucial for understanding our interactions with ecology and evolution in the Anthropocene. And this underscores just how, like with any other species, we have to consider humans and the societies we build to have diverse effects on the ecology and evolution of other species. So this is basically my call for interdisciplinary study of these dynamics among natural and social scientists, but perhaps most importantly, with communities of different people. Thank you.
All right. Fabulous recovery, Simone. It was almost like you planned to have that whole thing stall on the concept that humans are the ultimate ecosystem engineers.
That really set in, right?
Yeah, you planned that all the way. Anyway, from my point of view, a really profound talk. It gets my mind going off on so many different directions. And thank you for that call for truly interdisciplinary, transdisciplinary studies of this issue. I hope the awareness of the importance of this was raised for a number of people. So I would like to turn next to Marie McGraw, who is a postdoc in the Department of Atmospheric Sciences, where she has been for a year. Marie's work includes understanding and predicting extreme changes in Arctic sea ice on daily and weekly timescales. And as I'm sure she will allude to, this is of interest across the board to so many people, from scientists to policymakers. It's far away, but it's an extremely important effect. So Marie's presentation is entitled "Extreme Sea Ice Loss is Hard to Predict." I'm sure she'll convince us of this. Marie, are you ready to go?
I'm ready.
All right.
And actually, I ended up changing the title a little bit because it's really hard to talk about prediction in only 5 minutes. So Ellie talked a little bit about Arctic biology, and I'm going to take us back north to talk a little bit sort of about the physical environment of the Arctic, specifically sea ice, that frozen stuff on the ocean. And I'm part of the ice climate group in atmospheric science, and we talk about all things polar climate, not just sea ice. So if you ever want to talk about cold stuff, come drop us a line, hang out with us. So why do we care about sea ice? Sea ice helps keep the planet cool by reflecting sunlight, acting as a barrier between the ocean and the atmosphere. We've probably seen pictures of polar bears using ice to hunt. But most importantly, there are about 4 million people living in the Arctic across the world. And about 500,000 of these people are indigenous. And a lot of indigenous people live in small coastal villages, such as Kivalina, highlighted with a red pen or red pin on the map. And Kivalina is a small village on a barrier island in the Bering Sea. It's kind of famous for suing ExxonMobil over damages caused by climate change. And Kivalina, like many other coastal Arctic communities, people there use sea ice for activities like whaling and hunting. They use it for travel. And sea ice also provides this sort of protective layer from erosion caused by storms. So sea ice is really, really important for communities like Kivalina and all of the other similar communities in the Arctic every single day. And I highlighted Kivalina specifically because we've actually started working with leaders in this town specifically on research that's actually useful to people living in the Arctic. So scientists tend to focus a lot on changes in sea ice from year to year and decade to decade. Maybe some of you have seen a picture like this before. The orange line shows sort of average sea ice in the 80s, and the blue line shows average sea ice over the past 10 years. And you can see we had way more sea ice in the 80s than we do now. But sea ice doesn't just change on these sort of like longer, slower timescales. Just like weather, sea ice changes every single day. And also just like weather, sometimes the changes that occur on daily timescales can affect you more than sort of these big picture long-term changes, because that's what's happening to you right now where you are. So if you look at this little animation showing daily sea ice changes over the course of the month, especially in that red box, you'll see big changes in sea ice on day-to-day timescales. So we're losing a ton of sea ice over just the course of a week or two. And because sea ice influences and is influenced by weather, we want to use weather forecasting models to try to make better predictions of sea ice on day-to-day timescales. So people who live near and on the ice can sort of have this better information. So yeah, we want to understand these daily changes in sea ice better. So an example of the kinds of questions we're interested in asking are, what time of year do sort of the biggest changes in sea ice occur? And the answer is that it depends on where you are in the Arctic. In many areas, these sea ice loss events, especially these extreme events, the bottom 5th percentile, are actually the most common in the summer, though that's not true everywhere. Near Greenland, for example, they happen all the time. But a lot of you are probably sitting here thinking like, wait a minute, summer, there's 24 hours of sun. The ice is melting anyway. So of course there's more sea ice loss when you're already having sea ice loss. And you're right. So if we account for that and we, for example, compare like sea ice loss on December 2nd, 2020 to sea ice loss on December 2nd for the past 30 years, we actually see that we have a lot of extreme sea ice loss events occurring not in the summer, but in the fall. And fall is really, really important for sea ice. Fall is when sea ice grows, hunting starts in the fall, travel on sea ice can start in the fall. Those storms that can cause big erosion events on the coast, those are really common in the fall. So being able to make better predictions of sort of extreme sea ice loss events in the fall could be really, really useful for communities like Kivalina and other coastal Arctic communities where changes in sea ice from one day to the next can have a really, really big impact on people sort of living on the edge of the ice. So we're hoping that if we can understand these big sea ice loss events better and use tools like weather models to make better predictions, we can help communities like Kivalina and the other 400,000 to 500,000 Arctic coastal residents be more prepared for a future with possibly more extreme events. Thanks very much.
Awesome. Awesome, Mary. Thank you very, very much. I learned a lot from your presentation. And I loved how you combine all these changes in physical environment with the impact in human communities. It was really, really nice. Before I move to the next one, Simone, you killed it. It was great, really good. So the next speaker is Benjamin Miller. He works at the School of Environmental and Forest Sciences. He's a UDA postdoc for less than 1 year. His advisor is David Putman. And Ben's research seeks to identify the sources of fate of carbon in lake, river, and stream ecosystems. Ben's presentations is, do reservoirs make rivers more productive? Benjamin, are you ready?
Ready, Jose, thank you.
Go ahead.
So this is where I work, the Mekong River in Cambodia. Each year, monsoonal rains increase water levels in the Mekong in a pulse of flooding that expands across the landscape. Nowhere is this more apparent than Tonle Sap Lake, where I carried out my dissertation research with Gordon Holtgrave. The pulse of flooding that occurs in Tonle Sap Lake and elsewhere in the Mekong looks like this, expanding and contracting across the landscape each year. Water levels peak in October, as you can see on the y-axis of water depths. Upstream of Tonle Sap Lake, there is a new lake forming behind the Lower Cézanne II Dam, depicted here, where two tributaries of the Mekong meet. This is where I'm carrying out my postdoc research with David Butman. Like Tonle Sap Lake, this dam floods large amounts of land from before the dam's construction on the right to after its construction on the left. Unlike Tonle Sap Lake, this flooding of the landscape is more or less permanent, though it may fluctuate seasonally. Before the Lower Cézanne II Dam flooded the landscape, we think that the food web and fishery depicted here received carbon primarily from the upstream Mekong. This matters because carbon is the building block of all life. After the Lower Cézanne II Dam flooded the landscape, we think that dead trees and other land plants were broken down by microbes in the water at the base of the food web. When these microbes used all available oxygen, methane-producing microbes took over in the absence of oxygen. At the same time, we think that aquatic plants, such as macrophytes and periphyton, bloomed. Each of these aquatic plants provided the food web and fishery with still more carbon. So in the new reservoir, there are three possible major pathways for carbon fixation to the food web and fishery: photosynthesis carried out by land and aquatic plants, and methane production in the sediments. Because I wasn't around to study these carbon fixation pathways before the dam was built in 2014, I have to be content with studying them in the reservoir and comparing them to similar rates in its free-flowing tributaries, the Cézanne and Srepak rivers that flow into the reservoir. We haven't made it into the field yet due to COVID, but we hypothesize that the magnitudes of all three carbon fixation pathways will be higher within the reservoir than within these free-flowing tributaries. We further hypothesize that the reservoir has altered the dominant pathways for carbon fixation to the food web and fishery. In other words, we expect the relative importance of each pathway to be different within the reservoir and its tributaries. Each carbon fixation pathway can be traced in food webs using the ratio of carbon-13 to carbon-12, or stable isotope ratios. Here, in an idealized reservoir food web, you can see that photosynthesis by land plants and aquatic plants yields a different range of stable isotope ratios, as does methane production. We can use the stable isotope ratios in fish to determine whether they ultimately receive carbon from land or aquatic photosynthesis or methane production. We are interested in where these particular fish species receive their carbon. These species can be found during both the dry and the wet seasons in the reservoir tributaries, the Cézanne and Srepak rivers. We have stable isotope data for these species collected by others before the dam was built. Overall, we find that the stable isotope composition of these species shifts significantly from aquatic plants during the dry season to land plants during the wet season. This is true across feeding groups, whether the fish are insectivores, as pictured here during the dry season, omnivores, detritivores or bottom feeders, or herbivores or plant feeders. This means that whatever fish are eating, seasonal flooding drives the pathway for carbon fixation to the food web and fishery in this part of the Mekong. This implies that permanent flooding of land by the reservoir will as well. This is important to understand because dam building is increasing exponentially worldwide. Many of these dams are being built on some of the world's last free-flowing rivers, such as the Cambodian Mekong. Each of these is a planned or existing dam on the Mekong and its tributaries. If each dam changes pathways for carbon fixation to the food web and fishery, they could individually or collectively impact regional food security and food supplies. People in the Mekong have relied on fish since ancient times, as celebrated here on a mural from Angkor Wat in Cambodia. It's therefore vital that we understand the impact of dam building on this important food web and fishery.
Ben, that was really wonderful. And I really like the way you got us to think local to single river reservoir interaction out to that larger scale that you showed at the end in thinking about with a change in carbon fixation pathways, how we might really see differences, including in fisheries. Thank you. That was fantastic. Okay, we're now going to turn to Lisanne Kucha. She is a postdoc in the School of Aquatic and Fisheries Sciences, where she works with Sarah Converse and Beth Gardner. I am thinking you must also be in CEFS. Lisanne is an ecologist who specializes in analyses of carnivore space use and population dynamics, and how this information can help guide their conservation. And she will speak to us today on the recovery of wolves in Washington State. Are you ready?
I am so ready.
Excellent. Go for it, girl.
Okay. All right, so today I will be focusing on one of the most recognizable and polarizing mammal species on Earth, the gray wolf. Following the extirpation of wolves by the 1930s, the first Washington wolf pack was reestablished in 2008, and they continue to expand. So today I'll talk about the recovery of wolves in Washington and how it's not as straightforward as one may think, given that large carnivores are difficult to survey and there are a lot of moving pieces to estimate populations correctly. All right.
Are we wondering about advancing?
We are wondering.
Slides?
All right, there it is. Insights into wolf populations are especially timely given the Trump administration's recent delisting of the gray wolf from the Endangered Species Act, which removed federal protections, and the votes from the people of Colorado to reintroduce the species to their state. So the quotation here by Ben Maletsky really encapsulates the polarizing nature of wolves. Some members of the public welcome the reemergence of this species on the landscape, while others, like livestock owners, consider wolves a threat to their livelihoods. So given the current status of wolves is of great public interest, the big question is whether wolves have met statewide recovery goals. We can see here that wolves have been expanding on the landscape since their entry in 2008, but has it been enough? So the breakdown is that there are 3 recovery regions for wolves within Washington. As part of the state's recovery goals, there must be 4 breeding pairs within each of these regions over 3 consecutive years, and then a certain number of additional pairs anywhere in the state. So we really need to estimate numbers of wolf packs and individual wolves. And if you think, just go out there and count them, what's the big deal? You'll find that it's much easier said than done. One big thing that we have to consider when surveying for mammals is detectability. So detection is important to account for because in aerial surveys by fixed-wing aircraft or helicopter, you'll almost certainly miss wolves due to dense vegetation or fog cover, even if you know wolves are there because you're getting signals from GPS collars below. Another issue is that wolves live and die. Some have pups, some do not. Wolves enter the population and leave. And incorporating these processes of birth, death, immigration, and emigration are tantamount to properly capturing population dynamics. Insert the hero of this tale, the integrated population model. Without getting into the specifics, an integrated population model is really remarkable in the sense that multiple data streams on birth, death, counts can be incorporated into the same model. This model is still in development, but there have been some advancements thus far. First, focusing on the survival part or the death part, depending on how you look at it, we've used GPS collar data from 76 wolves to determine how long these wolves are staying alive. And we've found a couple things. First, the survival of dispersing wolves leaving the state drops to 0. And the dispersing wolves in general have the lowest annual survival, likely because of the foreign and often dangerous terrain they have to navigate. So, next up is the birth process. It's tricky because while there were pup counts on all packs when wolves first started recolonizing the state, and by pup counts, I mean there are visits to wolf dens to count the number of pups from the litter, this became more difficult as pack numbers increased. So what we will use are camera traps placed within wolf territories to try to quantify that aspect. While the camera traps were originally placed to get a picture of whether wolves were present, we're only interested in the number of young wolves in those images. As for the abundance side of the equation, how many wolves are on the landscape, we'll be using pack counts from aerial surveys. There are 8 wolves in this photo, and if you can't count them all, that means we do have to account for the fact that wolves are not detected perfectly. And thus far, we have not included any spatial element of this study. We can estimate births, survival, abundance, immigration without knowing exactly where those wolves are in space. However, that isn't sufficient given that the state's recovery goals are indeed spatial. And this process is hard to do because it's difficult to mechanize the tipping point at which wolves decided to recolonize the Northern Cascades and the Southeast. It's also difficult to predict this for future, oh, that happened fast. So we're working on the spatial extent for the model and building in the movement process. You can see here that wolves make long dispersal movements, this one into Canada, which are important in creating those opportunities for new pack territories. And one approach we're trying is saturating the state with hypothetical wolf territories and then essentially removing those packs that have low probability of wolf occurrence. Here's an example from the Northeast. The packs are in white. Maybe that bottom left one will be removed because wolves are unlikely to be there. So this is very much a work in progress, but once we have a model going, we can use it to not only inform current wolf recovery status, but also to simulate various management scenarios to project when recovery will be reached. It's all exciting, still in process. So thank you very much for your time.
Okay, now I got it. It was amazing, Lisanne. Thank you very much. We were talking about wolves and I was smiling the whole time. Pretty awesome, all the dispersal analysis that you folks did. It was impressive. And I was wondering, that's maybe a conversation that we can have later about the genetic diversity of that population of wolves in the state of Washington, if they are very related or they come from different families. But yeah, awesome. Loved it.
We'll chat later.
Yeah, thank you.
Thank you.
Next speaker is Pranab Roy Chaudhary. He works at the School of Environmental Forest Sciences. He's been a UW postdoc for a year now. He works with Dan Brown. And Pranab's presentation interests include land change modeling and urban science. Pranab's presentation, how sustainable are the forest management practices in the Pacific Northwest? Pranab, my friend, are you ready?
I'm ready, Jose. Thank you so much.
Go for it.
Good afternoon, everyone, and thank you for attending this talk, where I'll share insights from an ongoing work that explores the sustainability of forest management practices here in the Pacific Northwest and the role of relevant policy measures to ensure long-term sustainability of the forest lands. The Pacific Northwest forest not only supplies timber, but also helps sequester carbon, provide refuge to wildlife species such as northern spotted owl and the marble turrets, provide clean drinking water, and offer abundant recreational opportunities to the inhabitants of the land. Sustainable management of this forest can not only ensure the supply of goods and services, along with generating funds for public works, but it can also help us mitigate the impacts of climate change and help the local economy and environment in the long run. But the challenges to the sustainability comes in form of multiple landowners with their diverse management goals and practices, and this landowner's response to future market, policy, and environmental changes can actually have severe impacts that can tilt the sustainability. So we developed a computer model to simulate the harvesting behavior and their adaptation to future scenarios, and also a hypothetical carbon rent policy that pays the owners for the carbon in their stands in exchange of delaying harvest. Our model follows the socio-ecological system framework of Dr. Elinor Ostrom that consists of resource units, system, governance, and users. Similarly, our model consists of tree stands, also known as a group of trees, forest land, carbon rent, and other relevant policies, the landowners. The interaction among these components shape the outcome either towards or away from sustainability. In our model, we created entities to represent federal, state, industrial, and non-industrial or small landowners. These entities simulate the human behavior and their management action. We collectively call them the agents. While we assume the agents to be all financially motivated, the small landowners are unique because they value nature, recreation, conservation, and also the interaction with other small landowners in their neighborhood. And I'm going to focus on this particular segment in this talk. Our study area is located near the town of Forks in the Olympic Peninsula. And our synthetic forest inside the model is composed of land parcel, each of them owned by one of these agents, and 3 main tree species that are Douglas fir, western hemlock, and red alder. We ran this model to simulate a plausible range of market and policy scenarios, along with climate impacts on the forest. We then observed the response and adaptation of the landowner behavior to these changes, and we use carbon storage in their lands as a metric of sustainability over the next 100 years. So the landowning agents, they take multiple decisions a year, like which stand to harvest, when to harvest, whether the prices are good, whether they can delay harvest by a year, or get into the carbon rental market to earn more money over longer term horizons. But the small landowners are even unique. They also decide whether they value recreation or conservation over money and how their neighbors are doing. If they're also harvesting, then maybe this one particular landowner might be also inclined to harvest that year. This graph captures the carbon storage in the small landowners' forests under the baseline scenario as it is now, and the hypothetical scenario where they're being paid a supplemental carbon rent income to delay harvest. And surprisingly, we do not see much changes in the carbon storage outcome. So when we investigate further, we realize that the high upfront cost of entering such markets actually act as a hindrance to the small landowners, and that lowers their participation and also impact the anticipated carbon storage in their lands. So what do? We introduce an alternative choice to them by allowing the landowners to cooperate and form a large project by binding all their lands together. Now, they can collectively participate in the carbon market as one cooperative. And in the following graph, we see that the blue line shows the result of such cooperation. And we do notice that there is a significant amount of additional carbon storage under the cooperative scenario than the previous 2 scenarios. And we realized that by entering into a cooperation, the small landowners could lower this financial burden and gain competitive market advantage when compared to other landowners. This increased their participation in such carbon program and increased their carbon storage in their lands. So we realized that the management practices can vary widely among landowners, but with efficient use of relevant policies, such landowners can be empowered to embrace more sustainable land management practices. This work is still in progress, and we are still exploring various scenarios and outcomes. I thank you all for your time today, for being here and listening to my talk. Thank you so much.
All right, Pranab, thank you very much for opening up some new horizons, certainly for me. I just find it would take an enormous amount of courage and insight to combine in a single model social, political, natural variables. The complexity and insights required to do that sort of modeling is very impressive, but obviously very important for our region. So thank you very much for presenting that to us. Would like to now turn towards Marianne Reiss, and I think we're not doing too bad in time despite a few technical details. Marianne is in the School of Marine and Environmental Affairs and working with the Climate Impacts Group also. Her work focuses on helping communities prepare for impacts of climate change. And between SMEA and SIG, that is certainly the nexus of that sort of work in our college. I'm very much looking forward to what I think is the most intriguing title of the whole session. Her talk is entitled Transformation for Global Action, a New Type of Scientist. So Marianne, I'm looking forward to this. Are you ready?
I'm ready, thank you.
Okay, go for it.
All right. I'll be talking about my work training early career scientists at the Northwest Climate Adaptation Science Center, where our mission is to produce relevant science on climate change impacts and adaptation actions for Northwest natural resource managers and policymakers. So that means we develop science that communities can use to respond to climate impacts. For example, how to prepare forested communities from the impacts of increases in wildfire, how to restore habitat for salmon, or how to prepare coastal communities for sea level rise. And from our work with managers, we understand that one of the challenges communities face is not having access to science that is useful and usable to help prepare for the impacts of climate change, such as those I just described. And that's because science is often made in a way that's divorced from the complex context that managers face. This creates what's often referred to as the science management knowledge gap, which is the gap between the scientific knowledge and what knowledge is needed by communities and decision makers who are addressing climate and other challenges. And while many things need to be done to close this gap, one thing we need to do is transform the way we train scientists so that they have the skills and experience to work with communities to produce usable science. And so most science training follows the vision that a scientist works independently, gets an idea, conducts research, and then puts it out there into the world as a peer-reviewed publication. And we hope that a decision maker finds that paper and uses it to inform their decisions. In reality, this rarely works. A decision maker might never see it, or it may be totally irrelevant to the community's context. And unfortunately, most science training continues down this path. Instead, we need to train scientists that are capable of continual engagement with decision makers and communities throughout the science creation process, from collaboratively developing research questions and carrying out a project, as well as collaboratively sharing the results. And so this process can result in science that is more useful and used in decision making, including products that aren't just peer-reviewed papers, including things like tools for managers, collaborative networks, websites for sharing resources. And so for us at the Northwest Climate Adaptation Science Center, the goal is to develop scientists who are capable of collaboratively co-creating knowledge with societal partners or non-scientists. And this requires a new set of distinct skills that are not typically obtained in science training. Scientists need to be capable of working beyond their core discipline, including needing to understand complex systems thinking across sectors, strategic thinking around what knowledge is needed in climate planning and policy, and anticipatory thinking in a complex world. And while some scientists receive science communication training, it's often one-way science translation skills, while training for communication skills needed for multi-way communication is also needed, like listening and meeting facilitation. And scientists need to be capable of working with people from different communities representing different types of knowledge. And this requires that scientists can recognize different values surrounding their work, have interpersonal skills, as well as being comfortable with uncertainty and ambiguity. And lastly, scientists need to be able to interrogate their own biases and how they produce knowledge. They need to be able to identify and reflect on the values that their knowledge practices favor over other forms of knowledge so they can understand their position in a collaborative process. To summarize, scientists need a variety of skills to produce actionable climate science, and obtaining these skills requires experiential learning coupled with reflection activities. And we do this through our Northwest Climate Adaptation Science Center Research Fellowship Program. Our fellowship training augments traditional science training by funding graduate students and postdocs to collaborate with resource managers and communities on their science. We also provide training and support for collaborative research skills through a course, webinars, one-on-one coaching, and other activities. Scientists also need a community of practice to continue doing this type of research throughout their careers, and we work on developing this through peer-to-peer learning and connecting fellows to a network of scientists and resource managers as they launch their careers. So far we've had 3 cohorts with a total of 32 fellows spread across our 6 consortium university. And these fellows have worked with over 50 different organizations, including federal government agencies, state, tribal, and other entities. And these training activities provide a starting point with knowledge, tools, and skills. And we emphasize with the fellows the need for scientists to continue to practice, grow, and expand these skill sets throughout their career. And we've been evaluating the effectiveness of this training model using different metrics. And one fellow shared, this broadened my perspective in collaborative work and provided me with more tangible skills to approach transboundary research. It challenged me to think hard about my work as a scientist and inspired me to move forward in my career toward applied research that has meaning beyond scientific inquiry. Thank you.
Marianne, that was fantastic. I think you need a new bumper sticker which says, emotional intelligence is not an oxymoron.
There you go.
Right? I mean, so what you're talking about is boundary spanning and moving science to the next place. So man, snaps to you and that program. Thank you so much. Okay, so Anchor Space belongs to Max Stevens, now with NASA, but formerly a postdoc in Earth and Space Sciences with Ed Waddington and Michelle Kutnick. Max studies how snow on the top of ice sheets changes in time and how that affects sea level rise. And he will wrap it up for us today with measuring snow compaction at the South Pole. You ready, Max?
I think I'm ready.
Take it away.
Okay, so hi, I'm Max, and I'm excited to tell you about recent research I've done at the South Pole. And first, I need to give you some terminology. Firn is the old snow on top of glaciers that's compacting and turning into glacial ice. So the picture shows a bit of a firn core. The snow has been squished together, but it's not yet solid ice. So why am I studying firn? It turns out that understanding the firn is important for understanding mass change on the Greenland and Antarctic ice sheets. When the ice sheets lose mass, sea level rises. The images here give you an idea of the size of the polar ice sheets. If all of Greenland's ice melted, global sea level would rise by about 23 feet, and likewise it would rise 200 feet if all Antarctica's ice melted. The ice sheets are currently losing mass, and understanding how quickly they are losing mass is important for predicting future sea level rise. One way that we can estimate ice sheet mass change is by measuring the surface elevation using satellites. These satellites shoot a laser at the ice sheet, and by measuring how long it takes for the laser beam to bounce back to the satellite, we can determine the elevation of the ice sheet to within a few centimeters. The challenge is that the firn layer compacts and becomes denser as new snow falls on top, as illustrated in this cartoon. If we measure change in the surface elevation with the satellite, we need to know how much of that change is due to the ice sheet losing mass and how much of it is due to the firn compacting. For example, imagine that between January 2017 and January 2018, we observed that the surface elevation lowered. One interpretation of that measurement could be that the firn stayed the same and that the underlying ice thinned and thus contributed to sea level rise. Another interpretation could be that the surface elevation dropped due to the firn compacting. In this case, the thickness of the underlying ice is the same as it was before. Yet another interpretation could be that it's a combination of firn compaction and ice sheet mass loss. So how do we differentiate between these two scenarios? One way is to use a model, that is, an equation that predicts how fast the firn is compacting. The firn compaction rate is illustrated in the cartoon. It is the rate at which the two black lines, which represent the top and the bottom of the firn layer, which is about 50 to 100 meters, are getting closer together. We are confident that the temperature affects the firn compaction rate, but it is unknown by how much. So there are numerous firn compaction models that have been proposed by scientists. This plot shows predicted firn compaction rate at South Pole from two of those models. The x-axis is the date spanning 2 years, and the y-axis shows how fast the model predicts the firn is compacting. The two models are quite different from one another. The orange one predicts that the firn compaction is much faster in the summer than in the winter, and the blue one predicts that the compaction rate is nearly constant throughout the year. So, which one, if either, is correct? The key is to go out and actually measure firn compaction in the field. In December of 2016, I went to the South Pole to install instruments to measure firn compaction. We camped out for 3 weeks at a site 50 kilometers from South Pole Station. The pictures show South Pole Station and our field camp. At our site, we installed instruments in holes of different depths. Here's a cartoon of the experiment setup. We drill a hole and we anchor a string to the bottom with a weight. At the surface, there's an instrument called a potentiometer that measures how much string extends out of it. As time passes, the hole shortens due to firn compaction. The potentiometer has a reel that pulls in string as the hole shortens. A data logger keeps track of how much string the potentiometer reels in over time, which we can convert to the firn compaction rate. We also put a cable with thermometers in a hole to measure the firn's temperature. To give you an idea of what the process looks like, here's a time-lapse video of a colleague and me drilling a borehole. The tube pulls out a section of firn called a core, and we measure its density. Along with the temperature and the accumulation rate, density is an important control on the firn compaction rate. Here are some pictures of the experiment setup. The instruments are in wooden boxes mounted on a platform. The shallowest hole was about 4.5 meters and the deepest hole was about 106 meters. We had a box full of batteries to power our data loggers and solar panels and wind turbines to keep the batteries charged. When we left our site after installing the instruments, we had no clue if this was actually going to work. This method had been used elsewhere, but never at a location as extremely cold as South Pole. So we're quite pleased when we returned to collect our data to see that the experiment had worked. This figure shows the compaction rate we measured for the 2 years of our experiment. You can see that in the summer, when the fern is warmer, the compaction rate is about double what it is in the winter when the surface temperatures dip to about minus 70 degrees Celsius. The small wiggles correspond to warmer and colder weeks. We can compare our measurements in black to the models that I showed before. The orange model does a much better job than the blue model. Some previous research suggested that the fern compaction at a cold site like South Pole should not be very sensitive to the temperature, but our measurements demonstrate that it is. We can also use our measurements to create a new equation for fern compaction, which is now plotted here in red. It does a better job reproducing our data from South Pole. However, this is only one site, and our new equation might not work as well elsewhere. Our future work is to gather similar data at other locations. And I'll leave you a picture of the Antarctic ice sheet taken from the flight to South Pole. The measurements we made are important for improving our understanding of the fern atop the ice sheets, and we can apply this new knowledge to better understand how quickly ice sheets are losing mass and how they will change in the future. Thank you for listening, and I'm happy to answer your questions.
Max, that was great. You did a fabulous job of bringing us to the bottom of the world and the top at the same time. So, hey, hosts and postdocs, could you all turn your video and audios back on so everybody can see us? Wonderful. Thank you. I was just, I have to say, I was amazed. My now short hair was just blown back by the professionalism and the research diversity of approach, of system, of type of question, how we went from discovery science through to solution science, actionable science, just absolutely fantastic work. Bruce, Jose?
Yeah, that time went by really fast just because it was so intellectually engaging. And especially, I want to thank all of you postdocs for making your talks accessible to someone like me, a volcanologist who just stares at rocks. I actually got it all. So thank you very much. We understand this took extra effort on top of everything else you do, but I'm hoping that in return for this effort, there will be a sense of community. This will start to engage with other people. There will be further conversations. And maybe even the next time we do it face to face. That would be amazing.
Yeah, I think it was phenomenal. It was amazing. I'm fairly new at UW, so this allowed me to see what's going on in the college, what is the research going on, and the depth that the research is going forward. I thought it was amazing. So great job, you folks.
Yeah. And Max, you're doing double team. I hope you see you've got a question or so in the Q&A. So yeah, want to open it up a little bit to you 9. You just had one of the fastest hours of your life, I'm sure, both waiting to give your talk and then zooming through it. What do you think? Is this a good way for us to showcase forefront postdocs in the college? And just turn your mic on and speak up.
Yeah, it was fun to hear about things that weren't weather and climate, which is normally what I personally think about. Like, I never think about things that are alive, like fish or wolves. So that was pretty cool.
How about anybody else? Pranav, you look like you were going to say something.
Yeah, I think it was a great learning experience for us, and definitely it is as we think and we try to polish our work, which I think which then takes it to a stage where it's a great way of showcasing our work to the outside world.
Yeah. So Ben, you're a relatively new postdoc in the bunch. What do you think?
I think, I mean, it was a great way to learn about not only what everyone's doing, but also communication style. I think that all of us are constantly struggling to communicate our work more clearly, and it was great to see how everyone's accomplishing that. And yeah, I really admire the presentations by all of the other panelists for sure.
Yeah, and let me just say that you all are getting massive congratulations and great jobs, and 2 thumbs, 4 thumbs up in the chat. I can see, so obviously we had a very, very impressed audience. And I know that for you guys, it was not only just doing that presentation and like nailing it, but then having to do the Q&A and sort of think with your fingers at the same time. So, you know, little bit of a different thing. Bruce.
A question that I would like to put out to all of you who gave presentations, and you don't have to answer it here, maybe just emails to Julie and me. It was very professional, very smooth, wonderful slides. I don't know how much time you put into these talks developing, but obviously it was significant. And maybe you could give us some feedback if you're feeling that the time invested in doing this, after now you've done the whole process and maybe the hard work is a little bit in the distant mirror, was it worth it to you? Or to put it really simply, would you do this again on another subject? We're always looking to balance effort put in and what you get out of it, knowing that you are on soft money, you have a lot of pressures to produce your research, and this is something extra that we've asked you to do. So send us some comments via email if you care, but it's just also a way of me thanking you for taking that extra time and that effort to come up with a really engaging series of talks. Thank you very much.
Yeah, yeah, excellent.
I just want to add, I completely agree. That was a great comment, Bruce. I also want to highlight the fact that I saw a lot of names from undergrad students asking questions in the chat, in the Q&A. So besides your perception of how this work, keep in mind, it was a tremendous impact in our undergrad community. And I thought that was fabulous, because that can see themselves how there can be in a few years from now. So double thumbs up. I think it was phenomenal.
Excellent. So we are zooming to out of time. We have about a minute left. So one thing I want to say to all our attendees who are still with us, and there are quite a few, thank you, thank you. Thank you for coming. Thank you for putting your thoughts in the chat. Thank you for asking questions in the Q&A. And just the same way that Bruce put the invitation out to the postdocs at UC before you, who did these fabulous presentations, to interact with us, with Bruce and I in the Dean's Office, College of the Environment, about this event. Please, attendees, if you have thoughts about the next Pecha Kucha that you would like to see, postdocs or others, or other events that you would like to see that forefront the amazing science and scholarly work that we are doing in the college, please, please let us know. And we will do our utmost as a Dean's Office to put that together for you. So with that, many, many snaps and thanks out to the 9 postdocs who volunteered to lead us off on our very, very first inaugural Pecha Kucha of postdocs at the College of the Environment. Thank you. Snaps to all of you, and see you later.
Take care, friends.
Bye all.